Okay, so we are live, Antonia. Still waiting for people to join, but just a heads up that we are live already.
Okay.
Mhm. So, though the only thing that wait, actually, so let's see. Okay, hello, good afternoon, everyone. Thank you for being here. I know it's already a bit maybe dinner past dinner time, so but it's always a good time for speaking a little bit about butterflies. So we have Antonia Montero today here. She's joining us all the way from Singapore, and she's going to be talking to us a little bit about color patterns and how they appear in butterflies. So let's just give a couple more minutes for people to join in. Hello from YouTube as well. Either Zoom or YouTube. Uh, Feel free to leave comments in the chat, and we'll be happy to either tackle the questions during the talk or maybe at the end if we have time for the Q&A. But anyways, welcome, welcome. Just sit tight, and we will start in about a couple of minutes. There we go. let me just give the official intro again and start the recording.
Recording in progress.
There we go. So, again, good afternoon, everyone. Or evening. Thank you so much for being here, both in YouTube, the ones that join here in Zoom as well. We are so honored of having Dr. Antonia Montero, all the way from Singapore, joining us for this second day of talks for the Global Butterfly Week. So again, thank you so much, you all, for being here. She's gonna talk to us a little bit about the color and on butterflies, which is, I think, one of the things that capture us the most, at least at the very beginning when we are starting our journey with butterflies. So I'm very interested to learn about that, and I'm sure she will teach us some things. So, Dr. Antonia, thank you so much for being here, uh, and take it away.
Okay, thank you very much, Rodrigo.
Sure.
Okay, so today I thought I would just give you an overview of how we study why and how butterflies display their different color patterns in, for instance, dorsal and ventral surfaces, forewings and hindwings, and males and females. So there's all this variation across wings, surfaces, and sexes that we're trying to understand, both at the ultimate selective level and also at the proximate level. So let's see.
Whoops, I sort of need to move on. So there's all these butterflies have evolved ways of creating color on their wings using pigments, modifications of cuticle in the individual scales that make up like the color patterns on their wings that reflect light in specific ways. So we call that structural color. And there's a huge diversity of color patterns across all these surfaces and sex. So why, in terms of selective terms, why does wing pattern diversity evolve? I mean, for the many reasons. I mean, butterflies live in different habitats with different light properties, colors, different predator guilds in each habitat, where they can hide against tree trunks of different colors. They forage on different flower colors, etc. Find a mate, and then once they find a mate, they have to lay eggs to continue to propagate for the next generation. And so these are the two main things that butterflies are trying to achieve, and they use their wing patterns to achieve them. So wing patterns have these two functions. They have to somehow avoid predation, but also be appealing enough to attract a mate. And butterflies have been able to fulfill both these functions by partitioning where they put their patterns in more visible places or more hidden places to fulfill these two types of function. So for instance, when butterflies are at rest, mostly their ventral patterns are visible. And here this species has used this strategy called warning colors to prevent being eaten by predators. So they're signaling that their body has toxins, is distasteful, just like a mushroom is also sometimes this very bright red. So we've done experiments with this butterfly in Singapore. It's one of the Dahlias butterflies that lives here. And we've produced all these different paper models by modifying the bits of color and making them into a grayscale version, or removing the black altogether, and also the colors into grayscale. We placed these paper models in the forests in Singapore with a little bait, a live mealworm attached as the body. And then we measured who were the most predated models and the least predated models. And what we found was that the wild type pattern was the least predated of all these options here, and then followed by the red pattern, the yellow pattern, So 1, 2, and 3. And then the other ones were the most predated. So these bright reds and oranges seem to be important for deterring predation. And by the way, the larvae of these butterflies are eating some toxic plants, and they are storing these toxins in the body. And predators quickly learn to associate a distasteful taste of the butterfly's tissues with these colors. Other types of strategies. This is a moth that was found outside my hotel room in Malacca, actually in Malaysia, in the highlands. It's an amazing example of masquerading, where this moth is trying to is quite visible, its wing patterns. It's not trying to blend in, but it's trying to pretend it's something it is not. Like in this case, a fly that's feeding on bird poop. And potentially flies are not easy to catch by any old predator. And so these moths are probably advertising a sort of a unprofitability. So if you go chase after me, I'll be flying away before you get me. So don't even bother, that type of thing. Other butterflies use different strategies to hide. So this is crypsis. They're trying to blend in with the browns of the environment. Some people call this masquerading as well because they're masquerading as a dead leaf. So it could be either. And others use wing patterns like eye spots for deflection. From attacks of predators. So the eye spots are often positioned at the edges of the wing, away from the more vulnerable parts of the body, the head, the thorax, and so forth. And so I'll show you a video of a mantid attacking a butterfly, initially with very tiny eye spots and then with large eye spots, and you'll see the difference. So here's a wet season form of a Bicyclosanninana butterfly and a dry season form. And here's the dry season form attack. So the attack goes straight to the body, so the mantid can detect the body and attacks there. But here, the mantid is going to attack the edges of the wing. That was actually the attacked butterfly that you can see here on the left. And here's a slow motion. So the mantid gets distracted by these eye spot features on the wings of the butterfly and draws its attack to the wing margins. Here's a quantification of those types of attacks. So hindwings in green, most of the first attacks by mantids, when you have prominent eye spots visible on the wings, are towards the hindwings, and forewings nearly 20%, and then the rest of the body, the head, the thorax, the abdomen, these are the vulnerable parts of the body, get attacked the least. So that's one of the functions we think eye spots serve. Another student in Singapore, Sebastian, he tried out this other idea that had been around for a long time, whether eye spots, if they reach a certain size, like many moths have single eye spots in the centers of their wings, if they get to that size, could they prevent attacks altogether? So the predators would just not trust attacking such a big individual or object with such a large eye spot. So he put out these types of paper models in the Singapore forests with many eye spots, a single eye spot and a single large eye spot, and looked at predation. So number of attacks, meaning when did the mealworm, so all these models have a live mealworm, which is a beetle larva attached here with a little belt, a little paper belt. And so he goes back, he puts them in the forest with these little attachments with that wiggle in the wind. The mealworm also wiggles and draws attention. And so he puts them in the forest and he goes back every day to see when the mealworm is gone. And here what you see is attacks. So he sets up everyone, in this case, every one of these different wing patterns here at the bottom. He sets them up in equal numbers together with a control wing pattern that has no eye spots at all, just a band. And he looks at the proportion that gets eaten relative to the one without eye spots. And here you can see that in experiments 1 and 2, where the experimental models had many small eye spots or a single small eye spot, many small eye spots is actually quite visible to predators. So the mealworm gets attacked, but we don't see the live interactions because these are just paper models. But the point here is that these small eye spots don't seem to deter attacks. So attacks do happen in these paper models. But look what happened to these other paper models that have much larger eye spots. They are deterring attacks relative to the attacks that are happening to this individual without any eye spots. So it does seem to be true that if eye spots get to a certain size, they start to have this other function as deterrents. And then another additional function of eye spots is they seem to be involved in mate attraction. So here's a movie that just shows a type of manipulation we can do to test the role of eye spots in sexual signaling. I'm taking a butterfly, a male, out of a cage. I'm opening up its forewing, and I'm going to dab the dorsal surfaces of the eye spots with a plant compound that eliminates UV light from the centers of the eye spots. And so we did this type of manipulation with pretty much all the eye spots on the wings of this particular African butterfly. You can see that the UV signal is gone in this particular male. And then what I do is I'm putting 2 males in a cage with a female and trying to figure out who the female mates with first. So the males are going to approach the female. They're going to try and court her by rapidly opening and closing their wings and displaying their hidden eye spots on the dorsal surface. And the female then decides whether she wants to mate with him or not. And here's a successful mating. And it happens, we discover that these matings happen primarily with the males that have the UV signal. So the ones where the UV signal has been removed with this plant compound rutin, those males do not succeed in mating. So in this case, the hidden eye spots, the ones that the butterflies reserve on the insides of their wings, have a completely different function than the eye spots that are visible to predators. In South America, Central America, there are these beautiful monarch butterflies where the males went to the extreme, and females have them too, through correlations, to remove all eye spots from the dorsal surface, or maybe they never had them there, but they use these incredibly bright colors, these UV structural colors that are blue. And so they cover their whole dorsal surface with this amazing brilliance. And this is primarily, we believe, for mate attraction. And they just left the eye spots on the exposed surfaces of the wings for potentially predator deflection or predator attack prevention. So butterflies are deciding what surfaces to use for what function. And they do this in very intricate and interesting ways, and they dramatic differences between dorsal patterns and ventral patterns, because these seem to be able to be used for these different signaling functions that fulfill butterflies' sort of imperatives in life, which is survival and finding a mate. So how can they do this? How can butterflies produce different wing patterns in different wing surfaces? So I'll enter now into the field of developmental biology to explain how we sort of study these processes. Wings as a whole, we believe they are just a single there's a single genetic program written in the DNA of the butterfly on how to build a wing. But there's going to be genetic factors that are going to be specific to certain wings and to certain surfaces that can modify that ground plan program, that sort of wing building program that all butterflies have. Those genetic factors are going to tweak that ground program, and they're going to allow different wings to have different patterns and different surfaces to have slightly different patterns, and also different sexes to have different patterns. So here's an example of what we're going to talk about. So ventral and dorsal surfaces are being pasted together here for one species. So each species here has its ventral surface on the left, its dorsal surface on the right, just to show you how these wing patterns can be quite distinct between the hidden surfaces, which are the dorsal, and the exposed surfaces when the butterflies close their wings, which are the ventral surfaces. Here you can also see that forewings and hindwings can have different patterns as well. So the butterfly in the center has orange forewings and very blotchy orange and white hindwings. The one on the bottom left has eye spots only on the hindwings, but not on the forewings, and so forth. Then male and female differences can be striking as well. So females are shown on the left, the female pattern, males on the right. This is just one surface in this case where I'm showing here. But they can evolve very distinct patterns. Often the males have the brightest, shiniest patterns. They often are the ones that are trying to attract females. And sometimes females have more blotchiness, like in the bottom middle row, maybe to try to be more cryptic against the forest background and so forth. But sometimes females have also bright colors. In some species, females also use their colors to attract males. So how are these patterns controlled within a wing, and how are they altered across wing surfaces? I'll show you here an example of how we study this. So here's a caterpillar of this African butterfly by Cyclus aninana. It's a fifth instar. If we make little cuts on the sides of the body of this butterfly, we see that the wings are already developing inside. So they're very small little wing discs, about 1 millimeter across. And at this stage, wing patterns are already being set out by the genetic developmental programs in the body. In this stage here in the left bottom, this is the prepupal stage. So the caterpillar curves up, attaches itself to the plant, and the wings are growing exponentially inside the body. And then in the pupal stage, the wings come to the surface. So this is the wing triangular here, the part that I'm pointing to. You can see the eye spot centers in the species are small raised little domes of cells you can see from the outside. That's the center of the future eye spots. So all of these patterning processes are taking place starting in the caterpillar stage. They continue in the pupal stage and until the adult emerges. Here's what's happening in the larval stage. So this is a very, very tiny wing and it grows as you go from left to right. And here we're just staining the wing with two genes that are marking the beginning of the venation process that's taking place. So this is when the veins start invading the wing disc, is this green color. The trachea that will later grow into the space of the veins are at the base of the wing initially, and then they start growing and invading and so forth. The red gene in the bottom is marking all the intervein domains. So these genes are just setting up the pattern of vein and intervein. And at this moment, all the sectors of a wing seem to be marked by exactly the same type of signals, like this red signal. But that's going to change soon. So different genes start being expressed in different patterns as the wing is growing. So for instance, this gene green here starts to be expressed in the central cells of the wing, this other gene in just these two sectors of the wing. This blue gene is going to be expressed in the vein area, this purple gene intervein areas, this green gene at the bottom at the margin, purple and pink genes also at the margin, but with different stripes of different sizes. This red gene here, orange in the top, is expressed in just certain sectors of the wing. And then here we start to see the eye spots appearing, the eye spot centers in this wing with the expression of another gene. So there's going to start to have a lot of complexity happening. And this complexity is primarily driven by genes like these red and green genes that are positioning veins and intervein tissue that set out a series of other expression domains of other genes that are vein dependent or margin dependent and so forth. So there's a series of orthogonal signaling processes happening, some of them promoted by signaling from the margin, others promoted by signaling from the veins. And these things sometimes interact with each other, like in the case of eye spots. So signaling from the margin, as you can see here, this is a reaction diffusion model that is mimicking what's happening in a single sector bordered by veins on each side. So we're modeling here just the sector of one sector of the wing. And what we're saying might happen in the development of eye spot centers is that signals from the margin at the bottom here in orange, they diffuse inwards into the wing blade, but there are some boundary conditions on the two sides of each sector. And there is a morphogen, there is a gene that is expressed also throughout the wing epithelial. And these two in substances, these two chemicals interact with each other as these diffusions are happening that set up a stable group of cells that become separated from the surrounding cells and become differentiated into eye spot central cells. So this eye spot central cells here in orange are surrounded by a sea of another gene here in blue, that creates this differentiation. And so eye spot patterns, for instance, start off by having these central cells differentiate in the larval stages, but then these cells become very important signaling cells in themselves, and they are the ones who set up the rings of color around the central cells at later stages in development, the pupal stage. So here, for instance, we have a pupal wing of a bicyclothanian and a butterfly stained. So these colors, by the way, what they represent are we are doing a technique called hybridization chain reaction. And what we are localizing is the mRNA, the messenger RNA, that is produced through gene transcription. So when a gene is active, when a gene is on, it produces messenger RNA. And by designing a probe with complementary bases to this messenger RNA, we can detect, using a fluorescent tagged probe, we can detect where are the cells that are producing this messenger RNA. And so here we are tagging two different genes. We're tagging Spalt in black, in purple here, and we're tagging another gene called optics in cyan. And so we can see that the bands of this in the middle of the wing here, we call it the central symmetry system, are being tagged, or the edges of the bands are being tagged with optics, and the orange rings of the eye spots are being tagged with optics, and the black area of the eye spot is being tagged with Spalt. And so these genes, later, we've discovered, for instance, that both optics and Spalt are required for the production of orange scales and black scales, because if you disrupt these genes, you disrupt the production of those types of scales. So this is, in the end, what an eye spot would look like. You would have scales of different colors because early on they were expressing these different genes that are important genes to regulate the production of melanins in individual scales or the production of homochromes in the case of optics in individual scales. So the scales are individual cells that develop from the epidermal cells. They are sort of cuticular projections that grow out of the plane of the wing. And then later on in development, they either incorporate pigments or they develop these structural colors that produce unique reflections of light in unique wavelengths and colors. Now, this was just a little bit of background on what happens to a butterfly wing as it develops and the multiple processes of differentiating colors and patterns on one wing. And this is what we call the default wing program that is written in the genome of every butterfly. But now I'm going to tell you about the modifications that happen to this generic wing program that allow different wings and different surfaces and different sexes to tweak it and to allow the butterfly to do different things with these different surfaces. So the butterfly has these different genetic quadrants. There are there are genes that are expressed only in hindwings, for instance, that are able to differentiate hindwings from forewings. And then there's also genes that are expressed specifically in dorsal surfaces that allow dorsal surfaces to be tweaked differently from ventral surfaces. So for instance, the dorsal surface gene is something similar to what our own body. So if we move forward, we have our ventrum, our belly, that's our ventral surface. If we turn around, we have our dorsal surfaces. It's exactly the same type of thing with butterflies. In our hands, if we look at it, if we look at our nails, that'll be our dorsal surface. But if we look at our palms, that'll be our ventral surface. And there's a gene there, which is exactly the same gene as butterflies use. There's a gene that is able to differentiate dorsal from ventral and give us nails just on the dorsal surface of our hands, but no nails on the dorsal, on the ventral. So this exact same gene is present on the wings of butterflies on the dorsal surface exclusively. It's called Apterus A. And so it's only expressed in dorsal surfaces. It's here we are seeing a top-down version. We see the whole wing is colored purple. Apterus is present in every cell on the wing, but if we do a cross-section of the wing and we look at it in cross-section, we see on the right that Apterus is only present in one side of the wing, and that's the dorsal surface, the dorsal epidermis, not the ventral epidermis, okay? So this gene is super important to tweak that ground plan, that ground genetic program on how to pattern a wing and be able to modify it, modify the wing patterns that are going to be dorsal specific. So what does it do and how does it do it? Apterus A codes for this protein called Apterus A protein. And this protein is a regulatory protein. It itself binds DNA and it binds to regulate the expression of other multiple target genes. If we mutate this gene that codes for this Apterus protein using tools like CRISPR-Cas9, so if we make double-strand breaks on the gene Apterus and we disrupt it, what we observe is something interesting. So here I'm showing you what the wild type surface of a dorsal wing of Bicyclus aninana looks like. The ventral wild type surface, which is very different from the dorsal. So there's more eye spots, and there are these silver scales in males. And here's a crispant mutant. What this butterfly is, it's a male butterfly with patches of cells where the Apterus A mutation happened. So this individual was injected with these CRISPR-Cas9 gene editing tools as an embryo inside the egg. This editing process took place in some cells, but not all cells of a growing larva, a growing embryo. And so what we see here is just a patchwork of edited cells mixed in with non-edited cells. But we can very clearly see which are the edited cells. So for instance, all these cells here that show silver scales, these are the edited cells that are reverting to a ventral identity. So in the case of the butterfly generic program, the ventral identity seems to be the generic program. And Apterus A is this extra gene that is on top of that generic program that modifies it and allows the dorsal surface to be free of the sort of to remove the silver, the generic silver program of scale development here. It prevents it from taking place. So when you remove the Apterus A protein, this generic silver program appears, and you have silver scales developing. Also, Apterus A seems to be a repressor of eye spots because you have very few eye spots on the normal surface and you have a lot of eye spots on the ventral surface. But when you repress Apterus A, like in this case, then eye spots start to show through on the dorsal side, meaning that Apterus was a repressor of the eye spot program on the dorsal side of the wing. Without Apterus, the dorsal wing surfaces revert to the default ventral identity. So what we understand then is that Apterus A represses the silver scale development program on dorsal surfaces. It's a repressor of the program that produces this beautiful blue silver scales instead of the normal wild type brown scales. And it's also a repressor of the eye spot developmental program. So potentially it represses this reaction diffusion mechanism that I explained to you earlier, but we don't know exactly at what stage of that process does Apterus A come in to repress it. Now, another gene that is part of this genetic tweaking program that modifies the default program is UBX. So UBX is a Hox gene that is expressed just in hindwings, like you see here on the right. This is a larval hindwing colored cyan, that we are visualizing the presence of this UBX expression, mRNA. And the forewing, the front wing, does not have it. So again, it's just a gene that is present in one wing, but not the other wing. And it's present on both surfaces of that wing. And what we observed is that when we remove UBX, again with Apterus A, with CRISPR, we can start reverting the hindwing into a forewing. So we start seeing silver scales appearing on the hindwing, which only appear normally on the forewing here. And we start seeing like a big eye spot appear here that mimics the eye spot on the forewing. The wing shape is starting to change and so forth. So again, UBX here, it's mutated just in a mosaic fashion, but it's giving us this idea that this gene is required to modify a default forewing program into a specialized hindwing program. So the forewing program seems to be the default program, and UBX is there to create a novel wing program, which is the hindwing program, by tweaking a few things. And the things that it tweaks is, again, represses silver scale development over here on the posterior part of the wing, and maybe promotes some of these eye spots here from appearing in these sectors, because when you don't have UBX, these eye spots disappear, and promotes larger eye spots on the hind wing in this case. Sorry, it represses, the normal function is to make the eye spots smaller, so when you remove it, the eye spots get bigger. So again, these proteins, these UBX proteins, over the course of evolution, have been changing a forewing default program into a hindwing program by interfering with multiple processes of either scale, structural scale development, like repressing the silver scale program, and also in this case, repressing eye spots from certain wing cells, wing sectors in hindwing. So it's a gene that can have multiple functions, that can be used for tweaking the default forewing pattern in multiple ways in order to construct a completely novel hindwing pattern. I'm going to skip this slide. And then the another interesting thing, the final part that I'm just going to talk about, is sexes. So why can butterflies develop different color pattern, why can different sexes develop completely different color patterns? When they share primarily all the genes are pretty much the same. So except for the sex chromosomes. How do the butterflies use the information in their sex chromosomes to create different wing patterns? And the idea here is that in the sex determination pathway, there's multiple genes that are used to differentiate sex, to differentiate the gonads of the butterflies and so forth. And one of these genes is called double sex. So double sex is spliced differently in the bodies of males and females. So it's a gene that can be activated in both sexes, but when it's activated in both sexes, the protein that is produced in those cells comes out looking different. And the reason is that the gene is here. I'm depicting the gene double sex in the as a DNA sort of form. So you see the exons. The genes come in pieces in eukaryotic organisms. So you have to splice together this bit of DNA with this bit of DNA. And then males splice this bit of DNA with this other bit to create this green protein down here. But females, in their cells, in their body, they take this bit of DNA, they splice it with the second bit. But then they use these other bits of DNA to splice together to create a different protein that has these green and orange bits spliced together. It's a slightly larger protein. And so these proteins have different shapes. And because they have different shapes, they will be having different functions in the body. And so the idea here is that males and females end up with different proteins that can regulate the default genetic programs for wing pattern development differently. So they use these different proteins to tweak a bunch of different networks, a bunch of different regulatory programs in unique ways in order to produce male-specific patterns or female-specific patterns. It pretty much all lies in these two proteins that are slightly different in shape. These proteins are also regulatory proteins, meaning they bind DNA to activate or repress expression of target genes. So what we did here, Anupama in my lab, what she did was she targeted this common part of the gene of double sex with a CRISPR guide RNA, so to produce a double strand break in double sex and prevent the double sex protein, both in males and females, from being produced or expressed correctly at all. So here we have butterflies that do not have this double sex protein in their functioning in their wing. And so what does that do? So I'll show you first a few differential patterns in males and females, again in Bicyclus aninana. So the males are the ones that have silver scales. Females do not have silver scales on their forewings. Males have these hair pencils, which are modified scales that are used to diffuse pheromones during courtship to the antenna females to try to attract females to mate with them. They also have these little glands on the wing that are the pheromone producing glands, and the hair pencils are right next door to them to help diffuse the odors. Okay, so females don't have those traits. Now, we're going to look at the role of double sex by simply knocking it out. So we're trying to figure out what is the role of double sex in either the female bodies or the male bodies. And here's what we see. In a male double sex mutant, we see that the silver scales are gone. So what we conclude from this is that we conclude that the male version, the male splice version of double sex, meaning this green protein over here, the male splice version of double sex is required for the production, for the activation of the genetic program that produces silver scales and that is male specific. So without that specific male protein, these silver scales cannot develop. And that's what we see here in males. But look at what happens in females. In females that have been mutated for the double sex protein, they become male-like. So these females now develop these hair pencils on their wings, and they revert to this sort of male trait characteristic. And so what this tells us is that in females, the double sex protein is functioning as a repressor of a default program. So in this case, the default program for these butterflies is to produce hair pencils in both sexes. That's the default genetic program written in the DNA of the species. The default program says produce these hair pencils in both males and females, but females don't need these hair pencils. So they've now used their specific unique protein, which is this splice form that they themselves only have. They use this double sex female protein splice form to remove these hair pencils from their bodies. But when that protein is gone, they revert to the default program. And so do the males. So the default program, the genetic default program of a wing would be not to have any silver scales. But the males have used their unique protein to produce silver scales and to regulate their scale developmental program differently from females. So the function of the double sex male protein is to promote silver scale development in males, and the function of the double sex female version of the protein is to repress these modified scales, these hair scales in female bodies. So all these proteins that I've talked to you about, Apterus A, which is a dorsal specific protein, UBX, which is a hindwing specific protein, And double sex proteins, the male and the female version, they're all transcription factors, meaning they all bind DNA to regulate next door genes. So they bind regulatory regions of genes. And by doing so, they bend the DNA, they bring the DNA, the promoter or the enhancer, next to the sort of promoter of a gene, and they recruit RNA polymerases and they start the process of gene transcription. And so they are regulatory proteins. They can affect the expression of many, many downstream genes as long as these downstream genes have evolved binding sites for them. And so it's through the evolution of these binding sites that these proteins can become regulators of a variety of different genes in the genome to regulate patterns differently. So going back to the beginning, there is a generic, a default wing developmental program that's written in the genome of every butterfly species. That generic program is primarily a program that produces forewings and produces ventral surfaces, but then using Apterus A and using UBX, the dorsal surfaces can become slightly tweaked. The hindwings can become tweaked and become distinct from that default program. And by using the sex-specific protein double sex, males and females can also tweak that generic default program into male-specific development and female-specific development. And so, how do traits become restricted to specific sexes, specific wing surfaces, specific wings? Just to drive things home, we have initially homogeneous program across all four surfaces and wings in a butterfly. And here, for instance, we have double sex male protein repressing, let's say, this purple gene and repressing it in just the males on the left. So the females, the double sex male protein does not bind, and there is no double sex male version of the protein, first of all. So the double sex male version of the protein can now, for instance, repress this purple protein just in males but not in females. And that can create right away a dramatic change in wing patterns if, for instance, this purple protein is activating some melanin pathway program or pigmentation program. If, for instance, the Apterus A protein develops binding sites to this purple gene again, it can repress it in dorsal surfaces where it lives, but not in ventral surfaces. And so you evolve a completely different dorsal wing pattern from your ventral wing pattern by using this protein. And if we have UBX and UBX evolves this purple protein evolves binding sites for UBX, then if that connection happens, UBX can repress the purple protein in hindwings, can repress its expression in hindwings while keeping the expression of the protein in forewings intact. And with these three simple proteins, you can create all the diversity of wing patterns that you see in butterflies in nature. So in summary, they display, butterfly wing patterns display amazing complexity that is shaped by both natural and sexual selection. Those are the selective forces that drive these changes if they are beneficial for survival or for finding a mate. But this complexity is orchestrated by signaling cells along the wing margin, the veins, and other areas of the wing that define the position of signaling centers. There's reaction diffusion mechanisms taking place. And so there's a default mechanism for patterning the veins and the wing patterns. Obviously, the wing pattern programs are constantly evolving. Every species is evolving their own unique wing pattern. But at the same time, these evolving wing patterns are making use of these three different genetic factors in parallel. So as the default wing program is evolving in a species-specific way, these three proteins that I told you about are being integrated and also modifying that program in a surface and wing and sex specific way to produce even more diversity that is used by the butterflies to survive and reproduce. Okay, the work that I talked about is primarily the work of Joycelyn, Surya, Tirtha, Anupama, and Yuji, and other members of the lab that are no longer here present. And I thank you for your attention. Thank you. We cannot hear you, Rodrigo.
Okay, it's better like this, I think. Can you hear me now? Yeah.
Still cannot hear you.
Really? Okay, that's weird. Give me a second.
I don't know if other people can.
Okay, can you hear me now?
Oh, now I can hear.
Okay, sorry, I muted my microphone so I would not interrupt you, and then I couldn't make it work anymore. Anyways, Dr. Antonia, thank you so much. To be honest, I was in awe, and I didn't even realize that it only like already an hour went by. It's very very interesting, and so before I act selfishly and I send all my questions, I just want to ask if people in YouTube have any questions. We do have a couple of people logged in. We had actually a few that logged and then logged out, but the ones that are still around, any question? Okay, well, they decide. I do have some questions because, to be honest, I do find this quite fascinating, and it is more finding the that just these three genes can do so much is, to be honest, amazing, and but it kind of made me think when you gave the example of the person that is you see the person and then the wings, like, so does that mean that evolutionary speaking, like many species share those same type of genes?
Yeah, so these three genes are, we think, extremely conserved across insects in general, not just butterflies. So Apterus A is expressed in the dorsal surface, for instance, of Drosophila as well. So you can go as far back as Drosophila. And there's not been a lot of work outside in other insects, but if Drosophila has it, butterfly have it, it's likely to be very conserved across insects in general. The same thing for ultrabithorax, the hindwing specific gene. Flies have it, beetles have it, butterflies have it. So it's a sort of it's a Hox gene that is expressed in the third thoracic segment of all insects. Crustaceans, lobsters have it too in a particular area of their body. So it's always expressed in that middle region of the thorax. And so the traits that grow out of that segment have the gene expressed in it. So it's extremely conserved. And then the third, the double sex, is also a gene that we share. So we also have double sex, although we don't use it as much as butterflies, but we also use it partly in our sex determination process when we are developing our own gonads in humans. So it's also a very useful gene for determining, regulating sex and gonadal sex across most animals, actually. So they are super conserved. And we also, as I said, we use Apterus to differentiate dorsal and ventral patterns in our own body. So it can be conserved massive distances, evolutionary distances. So it's very cool that these genes are so conserved and so widespread across animals, and butterflies are using them
Yeah.
to regulate their default wing program. There's just one wing program. They are just using a couple of tweaks here, but it's not just a couple, it's many tweaks. I believe that over the course of evolution, many tweaks. These genes have hundreds of targets. They did not all evolve at the same time. So potentially all butterflies started off with same patterns all over their bodies. And then maybe over time, these genes tweaked here, tweak there, and they create this incredible diversity between surfaces and wings and sexes. So it's probably a very gradual process, but what's interesting is these genes being potentially. I mean, I'm assuming this is the case. There has to be more work done across other butterfly species. So we've done all of this work with one species. There's one more species that has been used for this type of work, Junonia senia, but very few others. And so it's important to repeat these types of experiments for us to really be sure that these are the genes that are doing all of it. So I do welcome other colleagues to continue exploring these types of questions in their own systems. Yeah.
That would be very interesting. And to be honest, I was just thinking, what about non species that don't have sexual dimorphism? Do they even use double sex?
Yeah, probably they don't. Yeah. There isn't, that almost doesn't never happen in the butterfly world, because there's always these sex specific traits in wings. The pheromones are often located in wings. Some of them are located in the abdomen of butterflies, but many, they have these androconial patches in wings, these modified scales they are on top of pheromone producing glands, live cells on the wing that are secreting pheromones. But yeah, if you have monomorphic patterns, these genes probably have little function there.
Amazing, amazing. Well, I mean, thank you so much. I learned a lot tonight, and I'm a fanatic of genetics, so like right down my alley. So thank you so much for your time, and
You're welcome.
for people that stick around till the end, thank you so much. Uh, they didn't put any questions down the chat, but if I get any questions, I'll make sure to send them your way so you can solve those questions. Other than
That's
that,
fine.
thank you so much for your time, and for
You're
people that are
welcome.
around, thank you so much for being part of Global Butterfly Week. We are just starting, and lots of things still coming. Uh, but so yeah, have a great rest of your your weekend, and yeah,
Okay.
have a great day. Take care.
Thanks, Rodrigo. Bye.
Bye bye.